Okay, so I had a question about positive and negative controls, and I thought I'd just make a video clarifying what they are. So, controls in general. Every experiment needs controls, because what you're trying to do in an experiment is get a measurement. And to know what that number, that measure means, you have to be able to compare it to something. Otherwise, it has no meaning. For example, when we say somebody that is 6'5" is tall, we're comparing that person to the average height in the population. That comparison is being made. That's what tall means. I mean, by itself, the length has no meaning. So in an experiment that you are trying to measure something, you have to be able to compare that measurement to something. Let's go back to my example of infecting with a virus. So I have my plate of cells, and I'm just going to write it very tiny. Just imagine that there are cells in here and some liquid right here. And I have another plate with the same cells, right? And this liquid. And I have another plate. I have a drug. And this drug, I believe, will stop the virus from infecting these cells. Now, how do I measure whether it is stopping the cells from being infected or not? And the way that you can do that is a couple of ways. One of them is once cells get infected, they will produce virus. So there's input virus, and then you're going to have output virus, meaning that these cells are making new virus. So if my drug is stopping the input virus from infecting the cells, I should get little to none output virus. Now, how do I do the proper controls? So let's say that this is my cells that I'm treating. So this is my treatment group. In another one, what I'll do is I'll have input virus. And again, I will measure output virus. But there's going to be no treatment. So no treatment. In this one on top, I'm not going to put any input virus, but I'm still going to measure for output virus. Now you might be asking yourself, why would you do such a thing? And in this case, no treatment also. So let me just number these. So this is group 1, group 2, group 3. So the reason why we have number 1, I need to see if there's some sort of baseline signal. Because I'm measuring output virus. I'm using some sort of molecular technique, some way to measure the output virus. And there could be some background noise. And I need to know what that is, what that level of background noise is. So that will basically be my bottom, my 0. So this is my 0. My 0%. Now, the group 3, there is no treatment. So this will be my 100%. Meaning that I am not affecting anything. So this should be the maximum. Now the treatment group, and I might have multiple treatment groups where I'm putting the drug at different levels. This would hopefully fall somewhere within this range of 0 to 100%. I mean, if it goes above 100%, then my drug is not actually helping to stop infectivity. It's actually increasing the infection rate, or the infectivity of the virus. So I need to have the 0 and this 100% so that I can measure how well the drug that I'm using is working. Now, number 1 would be my negative control in this case. So this is the negative control. And number 3 would be my positive control. Now, in some other scenarios, negative and positive control get kind of weird and different. It's really easy to see it with clinical trials, for example. The placebo group is the negative control. So you're always comparing how effective the treatment is compared to the placebo group. You can't really have a good positive control when you're dealing with human beings unless, and I'll get into what that unless is right now. So let's say you have a clinical trial. I'm testing a drug that is supposed to be good for treating some sort of cancer. It's a new drug. There's already a known standard of care. So there's a standard of care, some other drug. So we'll say experimental drug. And we have known standard of care. And of course, placebo group. Now the placebo group would be somewhat ethically questionable, because if people are sick, you don't want to really give them nothing. So we'll just have this in here, because probably in real life you couldn't do this anyway, have the placebo group. But we're just going to have it in here, okay? Just because we want to showcase the different types of controls that can be done, the difference between negative and a positive control. So in this case, the placebo group goes through the same regiment, meaning that they maybe go to the hospital, they sit down and they get the chemotherapy and etc. They're not getting the active ingredient of the drug. So they become the negative control, right? So the known standard of care is kind of the positive control. It's something to compare the experimental drug to, but this doesn't necessarily mean it's going to be 100%, because hopefully your experimental drug will work better than the known standard of care. So placebo is definitely your baseline. In the case of known standard of care, it's kind of a positive control, but it doesn't necessarily put the ceiling on the maximum. So you hopefully want your experimental drug to maybe fall or work better than the known standard of care overall. So sometimes the controls can't be neatly put into this is a negative and a positive control, but they are controls. They're meant to help judge whatever measurement you get from your experimental thing and put that in perspective of what it actually means. Because without that, it's just a number, a figure that has no meaning unless you can compare it to something. And it's very important in all these cases that all the groups are treated exactly the same. So there's not variability among the groups. So with the example that we had above here, of course, everything is being kept the same because I'm using the same cells, the same amount of input virus in 2 and 3 are being put in. I'm measuring the output virus exactly the same way, so I can easily compare between the groups. Okay, I hope this answers the question. If not, please ask in Discord or get in touch with me and I'll come up with other examples to explain this idea. Okay? Thank you, guys.